Hello and welcome to another Mr. Carter Science Special. Please don't forget to support the channel by liking and subscribing. In this video we're going to be looking at two very important molecules in your cells, DNA and RNA, and we're going to be comparing the structure of both of those molecules. So you may be aware that in the nucleus of every cell there is DNA found in a particular structure known as a chromosome. The DNA coils itself up around some proteins known as histones, and this tightly coiled structure becomes our chromosome. Within human cells we have 46 chromosomes or 23 pairs. But how would we describe the structure of this DNA molecule? We can see here in this diagram that it's beautifully illustrated the unique spiral shape that the DNA molecule has, and we call this a double helix. And you can see each backbone here is spiraling in a slightly different way, so they spiral and coil around each other in the double helix. But there's a lot more to this molecule than just that unique shape. So DNA, or deoxyribonucleic acid, has a very particular structure that you must be able to describe. The DNA double helix is formed from two sugar phosphate backbones with nucleotide bases held together by hydrogen bonds. The DNA molecule is made up of molecules known as nucleotides, and we can see a nucleotide here. We have the sugar, and in this case that sugar is a deoxyribose sugar. In fact, it's a very similar molecule to the ribose sugar. The only difference between the two sugars is that deoxyribose is missing one oxygen atom and ribose has that additional oxygen atom. Within this nucleotide we also have a small phosphate group, and we have a nitrogenous base, in this case cytosine. All three of those elements together are known as a nucleotide. And DNA is made up of molecules called nucleotides. Each nucleotide contains the phosphate group, the sugar group and the nitrogenous base. And there are four types of nitrogenous base: adenine, thymine, guanine and cytosine. As you can see from this diagram, the nucleotide bases can only bond to their complementary opposite. For example, adenine can only bond with thymine utilizing 2 hydrogen bonds, and guanine can only bond with cytosine using 3 hydrogen bonds. In the GCSE exams, it is useful for you to learn the names of the bases, but you can get away with just utilizing the letters A, T, C and G. When these bases join with their complementary opposite, we often refer to them as a base pair. You can also see on this diagram the sugar phosphate backbone, one going up and one going down to form the two strands on the DNA double helix molecule. Cells also contain a different kind of molecule. It's very similar to DNA, known as RNA. In this diagram here, you can see that the RNA is acting as a template for the base sequence during protein synthesis. In fact, there it is. There is the mRNA molecule. The RNA molecule can also take a slightly different form and fold in this particular way to form this single-stranded molecule known as tRNA. So we have two different RNA molecules, M for messenger RNA that acts as a template during protein synthesis, and T for transfer RNA, which is an amino acid carrier molecule and is utilized at the ribosome in the final stage of protein synthesis. So how does DNA compare to RNA? In this diagram in front of you, you can see that the RNA molecule is in fact messenger RNA. However, the same rules apply to the other form of RNA, transfer RNA. DNA is a double-stranded molecule. The sugar phosphate backbones of DNA contain the sugar deoxyribose, And along with the normal bases adenine, guanine, cytosine, DNA uses the base thymine. But when we look at RNA molecules, RNA is a single-stranded molecule. And in the sugar phosphate backbone, it contains the sugar ribose. And mRNA does not contain the base thymine. That base has been replaced by the base uracil. So in mRNA, uracil will complementarily bond to adenine during transcription in the transcription process as part of protein synthesis. So three big differences there. mRNA is single-stranded, contains the sugar ribose and uses uracil, compared to DNA being a double-stranded molecule containing the sugar deoxyribose and using the base thymine. I hope you've enjoyed watching this video. If you have, please comment below, let me know what you've learned, let me know what you like, and don't forget to like and subscribe to this channel. Thank you very much for watching.